So today we will talk about seed inhibition. So what is this actually? Actually, this the when the enzyme is controlled by its own product. Now, obviously, when you will read the statement, you are not able to understand this properly. Let me explain this you with the help of example. For example, is A. Rate which is A. Consider this: I'm adding this substrate into an enzyme. This is a enzyme. Really, this is a enzyme, and I am adding this enzyme into this, and this is basically this is the substrate, which is the A1. Right now, it has formed. Now it is known as enzyme substrate complex. Right, this is now known as enzyme substrate complex. Okay, basically, in some, like in our body, there are some enzymes with some products which are produced, like which are there is metabolic pathway. There is a kind of metabolic pathway which occurs in our body. So basically, what will happen when this enzyme will get to the product which will made, for example, the product which is made is B. Now, what will happen on B? There will be another enzyme, enzyme number E to react, and it will convert the product into C. Now, again on C, and other enzyme will react, for example, enzyme E3, which will convert this C in D, right? So here D is our final product. Here D is our final product. So basically, this whole thing is known as metabolic pathway. This whole is known as this whole is known as metabolic pathway, right? But what happens sometimes, like the product we wished has now become, and there is so many concentration, like the concentration of product has been increased. There is a lot of concentration of the product. Now what will happen, this product will come over here and it will join this product, like the concentration of the product has been increased now, right? What we wished, it has been done. Now what will happen, this final product will come to the enzyme. It will come to the enzyme and it will join to the allostric side. Now, I told you that in non-competitive inhibitor, the substrate joins, the inhibitor joins at the allostric side, which alters the change, which alters the shape of the active site. So when this product will now become a inhibitor. This product will become now inhibitor, and this product will join the first one enzyme at the allostric site, and it will change the shape. It will now change the shape, and now the enzyme will become like this, and substrate we are having is like this. Now the substrate can't be joined, right? The substrate can't be joined. It means now this B won't be formed. If B won't be formed, how other will be formed? It means the whole metabolic pathway will be disturbed, and by this the final product won't get created, because we are having a lot of. So when this final product is used, when it is utilized completely, then this product will detach from this elastic site, and again the X side will be maintained as it was, and then substrate will attach to it and it will combine to B, then B to C, and C to D. So this whole thing is known as metabolic pathway. So I hope this is clear to you. Now let's read the definition again. Let's read the definition again, which is when production of enzyme is controlled by its own product, right? So the production of the enzyme is controlled by its own product. Obviously, the final product is controlling the it's this reactivity. So basically this is known as feedback inhibition. So this example, this feedback inhibition is actually the example of reversible non-competitive inhibitor. So this example of reversible non-competitive inhibitor, right? So I hope this is clear to you, and with this our chapter has been